November 2002. Within the space of a few months, an unknown virus emerges in China. SARS contaminates 9,000 people and kills close to 800. Fear takes hold of entire populations, who appeal for divine clemency. Professor Yi Guan, a specialist in mutant influenza viruses, is in charge of the SARS file. He must act quickly, for many new cases appear each day. It seems that the virus is transmitted through air, so there is a risk of very rapid propagation. Professor Guan's mission: to isolate the virus, find its origin and its mode of transmission.
Southern China is an easy place to generate new infection emerging disease. The human population is the biggest in the world, and the animal population also biggest. The human and animal have close contact.
Civet cat farms are suspected of being one of the probable reservoirs of the SARS virus. These wild rodents, a prized delicacy for the Chinese, fetch high prices on the black market. In the markets, in the heart of the big cities, the close contact between humans and animals represents a time bomb for the propagation of mutant viruses such as SARS. A virus is a living dead organism lying in ambush for a host to squat. To reproduce, the virus must invade a living cell. The virus has one of the simplest anatomies. Its surface proteins enable it to fit a specific cell, as a key fits into a lock. Inside are all the ingredients necessary for reproduction. Its genetic material, made up of strands of DNA, and sometimes RNA as well. RNA viruses are the most dangerous, as they mutate most frequently. The SARS virus belongs to this high-risk category. Professor Guan carries out his investigations incognito, without official vehicle or white coat, so as not to frighten the population. Poultry farms are closely monitored, since they represent a vector for the transmission of the virus to humans. The avian flu alerts of 1991 and 1997 are still fresh in the minds of the Chinese. The SARS virus belongs to the coronavirus family, so named because of the crown-like capsule that encloses it. The SARS virus reproduces itself in 6 to 8 hours within the cell. It has the biggest genome of the RNA viruses, a fact which endows it with a high potential for mutation. At first, the SARS virus was shrouded in secrecy. Confronted with the risk of a worldwide pandemic, an impressive arsenal of measures was implemented, transforming the daily lives of the Chinese. While most of the Chinese population remained cloistered in their homes, Professor Yi Guan made more and more cross-border trips. Despite his priority status as a researcher, he still had to submit to the stringent checks in force. Masks became obligatory, and the hygiene regulations were draconian. China almost went into quarantine. In the country's major airports, thermal detectors scrutinized the crowds of passengers. Any individual with a temperature above normal would be intercepted and obliged to undergo a more detailed examination. Suspected febrile cases were noted in a register maintained by health monitoring agents. At the borders, health information forms had to be completed by all travelers. The propagation of the virus was halted, but Professor Yi Guan and his team remain vigilant.
This side. If the SARS are still in the wildlife, the human population is the same, the market system is the same, everything is the same, nothing changing. So the virus is definitely will come back. We just don't know when it will happen. It usually might happen more severe than the older one, since they want to survive.
The SARS epidemic revived the old demons of the 1918 Spanish influenza epidemic. For Jeffrey Tobenberger, there is no question of a similar hecatomb recurring. His objective is to reveal the secret of this lethal virus.
What we know now is that the influenza virus was probably breathed in by about every single person alive in 1918, but about a third to a half of them got sick, and of those, a very high percentage of people for influenza died.
The epidemic was even more deadly than the Great War. Between 50 and 100 million deaths in less than a year. Through lack of oxygen, the victims turned black and died within a few days, their lungs drowning in their own fluids. Close to 40% of the American soldiers who died in the First World War were in fact victims of influenza. But at that time, no one suspected that the cause was a virus. The US Armed Forces Institute of Pathology holds some surprising archives. There are millions of samples of tissues taken from victims of disease and preserved in tiny blocks of paraffin since 130 years. A true goldmine for Jeffrey Tobenberger in his hunt for the Spanish flu virus. He hopes to find a trace of the lethal virus here. The culprit responsible for Spanish influenza is finally unmasked. It's a virus. This microscopic invader breaks into a cell, it liberates its genetic material and reproduces in the cell nucleus. The virus thus transforms the cell into a factory capable of producing thousands of copies of itself, which in turn infect other cells. In 1995, several samples discovered in the tissue bank enabled Professor Tobenberger to begin sequencing the genome of the Spanish influenza virus. But this was not enough for producing the portrait of the killer virus. Luckily, Dr. Joanne Hultin entered the team.
And one day, a visitor, prominent virologist, mentioned that somebody ought to go up to Alaska and look in the frozen ground for victims from the 1918 flu pandemic. I knew when I heard that, this is for me.
For half a century, Joanne has hunted the virus in the frozen soil of Alaska.
The victim number 11 was really an amazing sight for me. And I was so happy when I found that. I knew that this is where genetic material from the 1918 influenza virus would be found. So here is a picture of the woman I found at 7 feet in the frozen ground in Alaska. She had died in the flu pandemic in November of 1918. And she was markedly mummified. And she had been obese in life, so that her internal organs were well preserved. And that is why I found such a good lung specimens for you.
So Johan, you probably don't recognize these, but this is the tissue samples that you sent us from the frozen samples in Alaska that we processed to look under the microscope.
But was there any danger in digging up such a lethal virus?
There is no safety risk in studying the 1918 virus as we've done it. We're obtaining tiny fragments of the virus from tissues of people who died of the influenza virus over 80 years ago, and there's no live virus left in these tissues.
Jeffrey Tobenberger and Joanne Hultin now occupy an important seat in the history of virology. After 10 years of efforts, the genome of the Spanish influenza virus has finally been completely sequenced. This colossal task doesn't explain why the virus killed so many humans. Its genome does not present any determinating mutations. The answer lies undoubtedly in the surface proteins. It is thought that the virus of the 1918 flu was originally a bird virus, in which the surface protein slightly changed, enough to enable it to infect human cells.
What we now think is that the 1918 virus emerged from a very unusual kind of bird influenza, and we think that it slowly adapted over a couple of years into humans. What we can't say is where in the world this event occurred.
Today, the risks of a new epidemic of lethal avian influenza are taken extremely seriously. Migratory birds, especially aquatic species, are among the main suspects. It is now known that they carry 15 subtypes of flu virus. To study the viruses harbored by birds, scientists regularly collect samples on the shores of the French Camargue. Philippe Sabatier is in charge of this mission. Early in the morning, Philippe has an appointment with travelers from Africa. The first arrivals are not the ones Philippe quite expected.
Donc incroyable, il est un peu fatigué.
Every season, 4 billion birds return to the European continent via the French Camargue. It is a highly privileged place for monitoring diseases transported by birds.
On pensait que les animaux avaient des virus particuliers, les hommes avaient leurs propres virus et chaque espèce possédait des virus propres à l'espèce. On s'est rendu compte dans l'histoire récente de la virologie que certains virus pouvaient passer d'une espèce à l'autre indifféremment.
Bien rouge.
Philip has a special interest in viruses that are capable of moving from bird species to other animals or man. In the French Camargue, the conditions for this are present. Horses and cattle occupy the same territory as the migratory birds. With local farmers, Philippe Sabatier has established a monitoring system which makes possible the immediate detection of viruses of avian origin in intermediate species such as horses.
Le but du travail qu'on fait, c'est de mieux comprendre les différents risques, de façon à ce que on puisse déclencher plus vite des réactions pour éviter que ça se propage.
Fortunately, the species barrier is not crossed so easily. A virus becomes contagious when the temperature, acidity, and genetic and protein environments are favorable. To colonize a different host, the virus must adapt itself to the new conditions by mutating. What scientists fear most is that a virus of animal origin, such as the avian flu virus, and a virus of human origin infect the same cell. Their genetic material would then combine to produce thousands of hybrid viruses. These mutants, equipped with new camouflage systems and new weapons, could become true killers in humans. Philippe Sabatier also tracks viruses by satellite, thanks to the expert eye of Antonio Guel.
Les satellites d'observation de la Terre permettent d'étudier l'environnement, la végétation, la terre, l'eau, qui sont des facteurs qui permettent au virus de se développer.
The target? To prevent catastrophic science fiction scenarios from becoming reality.
What the hell are you doing? Turn off the oxygen. Put him on room air.
No, the air is thick with Andromeda. Experiment with your own life, damn it.
Andromeda will spread everywhere. They'll never be rid of it.
There are now 5 minutes to self-destruct.
On revient du terrain avec des prélèvements
Il y en a probablement des positifs. On envoie ça chez Pasteur pour l'isolement du virus. Est-ce que toi, tu as des informations par le satellite qui nous permettraient d'imaginer un risque d'épidémie aujourd'hui?
Antonia Guel works with Philip within the framework of the BIRD program, a worldwide study of the viral risks linked to bird migration. With micro-emitters attached to their bodies, the bird's most secret movements cannot escape the eye of the satellite.
On a reçu les dernières données d'émission des hérons. Il y en a qui sont déjà arrivés vers la Mauritanie.
With a precision of several meters, the bird's final destinations and all their stopovers are revealed. In the French Camargue, Philip divides the area into zones, in which he records the bird species present and estimates the size of the different colonies. Philip double-checks the information provided by the satellite through an aerial view. This year, the abundance of vegetation increases the risk of an epidemic, since the birds are likely to remain longer to take advantage of the luxuriance. Geese may not be very cooperative, but they are one of Philip's latest priorities. He would like to know more about their migration pattern. His first task is to familiarize the big birds with rudimentary attachments.
Donc c'est bon, on la lâche.
The excess baggage is not always appreciated by the geese. The risk of a pandemic is associated with highly contagious viruses that are transmitted through air. Like SARS and influenza, smallpox belongs to this family. Swapan is a miracle survivor of this terrible disease. He caught it when he was a young boy. Every evening, he praises Sitala Mata, the goddess of smallpox, for having spared his life. He reverently prepares his offerings, which he takes to the closest temple to celebrate the goddess with hundreds of other worshippers. The great pandemics were often interpreted as a divine punishment, an evil that was impossible to fight and which must be accepted with resignation. India was one of the countries most seriously affected by smallpox. Until the first successes of modern virology, the only remedy was to put oneself in the hands of the gods. This is why in India, the Hindu pantheon includes a goddess of smallpox, Sitala Mata.
সেই সময় স্মলপক্স প্রচুর দেহের মধ্যে হলে কলা পাতার উপর শুয়ে রাখা হতো, কারণ জামাগুলো সব আটকে যেতে বলে কোনো জামা কাপড় পরানো হতো না, পুরোপুরি সে উলঙ্গন মতো করে কলা পাতায় শুয়ে রাখা হতো। এটা আমি অনেকের দেখেছি।
Those who survived were left with terrible scars. Compared to religion and popular beliefs, Dr. Sarkar is the embodiment of science. A pioneer of virology in India, he created the first major laboratory in Calcutta. Dr. Sarkar directed the national vaccination campaign. He remembers how it was necessary to counter the resistance and beliefs of certain villagers who did not want to be vaccinated.
So I just went there, gave a strong lecture, just as an orator. What do you think we have come for? Not for myself, it's for you. You are being protected and all those things. And then, after lecturing, they just agreed.
In 1966, a worldwide smallpox eradication campaign was launched. India was among the last 25 countries affected by the disease. Smallpox was eradicated in 1979. The history of smallpox vaccination dates back to the 10th century. The Chinese immunized themselves with a powder made from the dried crusts of the sores of smallpox sufferers. In Europe, the powder was administered by skin puncture. This method was known as variolation. While smallpox may be the most deadly virus of the 20th century, there are other microscopic killers that hold impressive numbers of victims. In the war against viruses, man is trying to build up an efficient arsenal of defense. A race against time has truly begun. Each mutation is like a new secret weapon against which we must learn to defend ourselves. For more than a century, modern virology has been taking giant steps forward in vaccine research. But to protect humans efficiently, stocks of defensive weapons must be manufactured in sufficient quantities. The industrial war machine for vaccine production must be launched to put a stop to the invisible invaders. The vaccine must then be distributed to the four corners of the globe. Unlike vaccines, viruses travel alone. Vaccines reinforce our body's defense. The vaccine uses the properties of our immune system, capable of recognizing an invader. Sentinel cells tell the white cells the identity of the intruder to be surrounded and attacked. The white cells produce antibodies that fix themselves to the surface proteins of the virus. The enemy is neutralized and can no longer attack the cells. The invading virus is then killed by other white cells. Vaccine manufacturers were the first to create prototype high-security laboratories, now known by the code of Biosafety Level 4. It is here that researchers, like astronauts of the infinitely small, dissect active viruses. To combat an enemy, you have to know what weapons he bears. Viruses can infiltrate concrete and rubber, but through materials developed for nuclear applications, humans can now safely accept the challenge. The high security zone where viruses in their most dangerous forms are manipulated is an airtight chamber maintained at constant low pressure. For the researchers, there is an obligatory chemical shower at the exit. These laboratories of the fourth dimension now exist on every continent. There are deadly viruses, however, for which there are no vaccines. This is the case for hemorrhagic fevers such as Ebola. In the villages of Africa, the simultaneous arrival of this devastating illness and foreign medical teams, nicknamed the astronauts by local population, arouses confusion and mistrust. In the locals' eyes, these emergency teams are there only to disinfect the area, isolate the sick, and evacuate the bodies. Dr. Eric Leroy is called out on the first indications of an outbreak.
On essaye de comprendre pourquoi certaines personnes guérissent de l'infection, 20% plus précisément, et pourquoi 80% des personnes décèdent. Donc on essaye de comparer les réponses immunitaires de ces deux catégories de personnes.
Ebola is a filamentous virus, hence its name, Filoviridae, which first of all attacks the cells of the immune system, then the cells that line the blood vessels, thereby causing hemorrhages. In the field, the priority is to inform the population and to convince people to cooperate with the doctors.
Ebola ezalaki na kele. Tena mbomo. Ebola esili koyaka awa na kati na mboka. Lekolo yango eyi na kati na mboka. Nakobanga Ebola na kati na mboka.
Animal reservoirs carry the virus without getting sick. They transmit it to intermediate species, porcupines, antelopes and apes, that are eaten by the villagers. Humans are contaminated when they manipulate the meat of infected animals.
Actuellement, un certain nombre d'animaux réservoirs sont suspectés d'être porteurs du virus. Bon, ça peut être soit un petit mammifère, une musaraigne, une souris, ou alors une espèce volante comme un oiseau, une chauve-souris. Si on met en place un réseau qui détecte rapidement les mortalités animales, avant l'apparition des premiers cas humains, on peut mettre en place une série de mesures afin d'essayer d'éviter le passage à l'homme.
Ebola is transmitted through simple physical contact with victims of the virus or their body fluids. Infection is rapid and devastating. The sick die within several days. Volunteers recruited in the villages bury the dead under extremely cautious conditions. The areas hit by the virus are isolated to break the chain of contamination.
Est-ce que vous savez que la zone était en quarantaine avant que vous ne partiez à...
Oui, je savais ça.
Mais pourquoi vous n'êtes pas venu nous voir? Nous voulons sauver la vie de tout le monde. Hein? Parce que toi, si tu es malade, tu arrives à Brazzaville. Brazzaville, c'est une grande ville. Dès que tu es malade là-bas, c'est fini, Brazzaville tremble.
Les lunettes ici, dernièrement je vous parlais de l'étanchéité. C'était pas étanche et ça a coulé jusqu'à l'œil. Donc, il est mort. Donc, il ne faut même pas attendre qu'il y ait une goutte de sang, de sang, de vomissement sur toi. Ça, il ne faut pas des coups de diarrhée. C'est pour cela, tout à l'heure, nous parlons de l'habillage.
Les gants là, c'est écrit left to right. Hein? Gauche, droite. C'est en anglais.
The nursing teams must be shown how to protect themselves while caring for patients, for it is often the medical volunteers who are amongst the first victims of the virus. After use, all protective clothing is incinerated. For even a virus as deadly as Ebola cannot resist fire. By penetrating deeper into the forest, humans put themselves in contact with these viruses.
Over half of the face of the landmass of the earth have been changed by what humans do. That is not a trivial impact. We are a major player in the emergence of these diseases and in the changes in the ecology of the world. So we are our own worst enemy in this respect.
Professor C.J. Peters is the father of virus hunters. It was he who unmasked a new microscopic enemy called Nipah, which appeared in Malaysia in 2001. The virus was transmitted to humans from pigs that were suffering from a serious respiratory illness, initially infecting people who worked on pig farms. To prevent the propagation of the virus, the counterattack was rapid. Over 1 million pigs were slaughtered. Pig farms were closed and disinfected. It seems certain now that bats are the animal reservoir for the Nipah virus. Bats transmitted the virus to pigs, which in turn infected humans. What is more worrying is that the virus has also infected dogs and cats. It is therefore able to cross the barriers between several species. This is why C.J. Peters classifies Nipah as one of the most frightening viruses of recent years. Only these unknown viruses can get through security checks without ID cards.
Hi, excuse me, can I help you?
New virus from Asia.
Okay,
I'm Dr. Peters.
Dr. Peters, can I see if you got access to this building, please?
You know, people always ask you, what's the big one? What's the bad one? What wakes you up in the middle of the night with sweats and so on? And the fact is, in my experience, you don't know what the next bad one is. You can tell people, though, that the thing that's the most worrisome is a virus that can spread from person to person. And that is the virus group that we're most concerned with. But this one, there's not much data on. You know, when things happen overseas, there's not a very good surveillance network, and we don't have good setup for studying the details of what's happening. It really, I think, puts us at risk.
Every 2 or 3 years, the world is confronted with the same phenomenon. A new virus, totally unknown, attacks humans. Nobody knows where it comes from, how it transmits, or the size of the epidemic it may cause.
We will have millions and millions of deaths, and we will have a huge impact on, not just on the people who die and the people who are sick, but it'll change the nature of hospital practice. It will have a severe economic impact. People won't want to get on airplanes because that's a chamber where the viruses can circulate. People will not have the manpower to harvest crops.
Over 2,000 viral species live upon our planet, but only 10% of them attack man. After dissection in the BSL-4 lab, viruses are inactivated. They can then be studied without special protection and without the risk of contamination for humans. The moment when the virus reveals its true nature is one of anxiousness.
That's very interesting. That looks like SARS to me.
Yeah, it does.
Yeah. Let's take a look at the micrograph and see if we're sure.
Look here.
C.J. Peters has been called into action in all the great crusades against viruses, including the battle against SARS. This was not without sacrifice.
I wore a beard for about 20 years. Then in June of 2003, I was going to Taiwan to work with them on the SARS outbreak. And I realized I needed to have a respirator that would fit my face very tightly so that it would keep out the particles and protect me. So the beard had to come off.
C.J. Peters no longer works in the field, but others continue the virus hunt. Luis Eloy Pereira and his team have been notified of a victim of an unknown illness. The investigation is launched. They collect the clues that could put them on the trail of the killer. Luis Eloy is a biologist. He and his team relentlessly scour the caves, fields, forests and swamps of Brazil in search of viruses.
Renato, esse é morcego vampiro, porque ele é hematófago, mas ele também é reservatório de arbovírus, porque ele vive no ambiente silvestre.
Brazil has a long tradition of virus hunting. Its climate and its wealth of flora and fauna are factors that favor the development of certain viruses, especially the arboviruses. This term designates all viruses transmitted by blood-sucking insects. Bats and rodents are favorite reservoirs. That's why these little animals are the favorite subjects of research for Louis. Of the world's 537 known arboviruses, 400 have been detected in Brazil.
Ô Renato, esse é aquele exemplar que nós capturamos, que detectamos arbovírus, lembra? Eu vou colocar na estante para identificar. Tá legal?
Louis' interest is not only in the viruses themselves, but also the risk of propagation. In this region of the world, birds are among the species that are closely monitored. It is important to know that, like rodents, they can be carriers of arboviruses. For thousands of birds, Brazil is an unavoidable migration corridor between the northern and southern hemispheres. One of Louis' fears is that birds might transport viruses picked up in Brazil through being bitten by mosquitoes or other insects to other countries. Recently, separated by a distance of 12,600 kilometers, the same virus was isolated from a human and a bird.
Eu venho há 35 anos caçando vírus. Eu e minha equipe conseguimos isolar 25 vírus novos para a ciência, como o Rocio, o São Luís. Mas esta paixão é muito perigosa, pois uma vez eu me contaminei com o vírus Guaratuba, quase morri.
Rocio is one of the viruses that worry Louis most. It has been found only in Brazil, unmasked for the first time in 1976 in a person who died of encephalitis in the city of Iguape.
Essas telas, Jonas, contêm os vírus que nós encontramos nos morcegos já capturados em Iguape.
Every year, Louis Eloy's Institute tests 30,000 samples collected from animals or humans. The invention of electron microscopy in 1939, a technique which can magnify 30 to 50,000 times, offered scientists the possibility of examining viruses. Up until that time, the most that could be seen with optical microscopes were large bacteria. But even with an electron microscope, viruses are so small, the viral surface proteins appear only as vague outlines. As opposed to the optical microscope, which functions with light, the electron microscope uses a beam of electrons which sweep over the specimen to be examined with great precision. This type of microscope allows us to go much closer in our observation of the infinitely small. An average hair measures 1/100th of a millimeter, a mold spore 3/1000ths, a bacterium 1/1000th. The virus world begins at 1/10,000th of a millimeter with the smallpox virus, one of the largest, then the coronaviruses, and lastly the rhinoviruses, the smallest, measuring close to 100,000th of a millimeter. For John-Yves Gros, this wall of numbers has a true meaning. They are the measurements of a virus in 3 dimensions. To the trained eye, these figures reveal the first secrets of an unknown galaxy. In answer to the challenge that viruses present to us from their invisible world, John-Yves' weapon is computer technology. Like a surveyor, he measures the contours of the viral envelope. His goal is to produce 3-dimensional models and reproduce exact replicas of the original viruses.
The very first time I had an image of a virus on the screen, I was really excited. Suddenly, from all these bunch of numbers that wouldn't normally mean much to anyone, came to life this new world where no one had gone before. These new worlds with mountains, valleys, canyons that could be mapped into a three-dimensional atlas for all these viruses.
The technique developed by Jean-Yves Gros reveals the identity of the virus and provides extremely precise information about their shapes and their surface structures. A great step forward to a better understanding of their weapons of attack. But the great virus family has more surprises in store, for there are still unknown entities, aliens that emerge from goodness knows where. This is the case of mimivirus, the only virus large enough to be observed today with an optical microscope. Extracted from the air conditioning circuit of a British hospital in 1992, this thing, for a long time considered to be a bacterium, has resisted all scientific investigations for over 10 years. In 2003, Didier Raoult received a specimen for analysis.
La première fois qu'on a vu ça, on était sidérés parce qu'on n'attendait pas du tout cette forme. On s'est retrouvé face à une forme très géométrique qu'on ne voit jamais chez les bactéries, et on a mis du temps à réaliser parce qu'il y a une espèce de blocage qui empêchait de penser qu'un virus puisse être aussi gros que ça. C'était impossible. On voit bien là que quand on met mimivirus à côté d'un certain nombre de bactéries, en particulier les plus petites, il a un diamètre qui non seulement est comparable, mais parfois plus grand qu'une bactérie, et on comprend qu'on ait pu le confondre pendant des années avec une bactérie.
The sequencing of its genome will no doubt reveal other surprises. 80% of the mimivirus genes are totally inexistent elsewhere. On a microscopic scale, it's a bit like finding a living dinosaur.
Ce que mimivirus nous apprend, c'est que nous savons très peu de choses sur les virus. Il y a plus de virus que n'importe quelle autre forme de vie sur cette Terre. Il y a des formes extrêmement curieuses de virus qui ont été aperçues au fond des mers, dans les boues, dans l'eau. Et globalement, le monde des virus est un monde à découvrir.
If mimivirus is unique and has never been found elsewhere, there are other viruses that proliferate throughout the natural world. This New Yorker is fishing for bacteriophage viruses, literally bacteria eaters.
If you line up all the bacteriophage that are present on Earth end to end, they would extend 200 million light years. What that means is if you traveled at the speed of light, it would take you 200 million years to get to the top of the stack.
Vincent Vichetti uses these viruses in the war against pathogenic bacteria. The effect of bacteriophages is devastating. Their action turns a slightly opaque bacterial solution transparent in just a few seconds. Anthrax bacteria literally explode under the effect of bacteriophage.
Half the number of bacteria on Earth turn over every 2 days because bacteriophage kill them.
This animated sequence, inspired by electron microscope images, quite faithfully reproduces the structure of the virus and how it reacts. As soon as a target bacterium enters its environment, the virus attaches itself to the bacterial cell membrane, then injects its genetic material into it, using a sort of microscopic syringe. The bacteriophage multiplies inside its victim and then explodes it. Bacteriophages are everywhere, in the smallest drop of water or a handful of soil, and Vincent Vichetti does not have to go far to find his microscopic companions. For him, Central Park is a gold mine.
It's more than we've ever been able to do before, because right now we wait for the infection to happen and we treat with antibiotics. This is our first opportunity that we can prevent infection. Other than vaccines, this is the only other way you can go in and prevent infection, by removing the pathogenic organism or the disease organism before it has a chance to cause infection.
Although most of the research funds allocated to Professor Fischetti go to anthrax research, he has also begun to develop some products that hold great promise for everyday medicine. He will soon be seeking market approval for aerosols to combat pulmonary infections. Daniel has formed an alliance with the cold virus to attack a terrible enemy, cancer. Dr. Daniel Sees has experimented with an engineered cold virus against liver cancer. The results are very encouraging. Injected into the artery leading to the liver, the virus exterminated tumor cells without damaging healthy tissues.
There was a woman in the early 1900s who had cervical cancer and then caught influenza, a severe case of flu, and her cancer got better. And people noted it but weren't really sure why. But it's been in the back of many people's minds for decades and for generations that maybe we can use viruses as a weapon against cancers.
With Dr. Sees, the cold virus entered the hospital through the front door.
Patients that we expected to live only a few months, on average, overall lived about 1 more year. It's not a miracle. Most of these patients ended up dying of cancer, but we were able to extend their lives and buy them a little time. So here we have CT images of a patient who was treated on the Onyx virus trial. At the time of presentation, he had colon cancer that had already spread to the liver, and you can see that there are multiple dark areas within the liver that represent these tumors. After being treated with the Onyx virus, 7 months later, we performed a follow-up CT scan that revealed that most of these tumors have completely disappeared.
Research bringing hope in curing a disease that touches 16 million people around the world. We are not dealing with Dr. Jekyll and Mr. Hyde, but with recognized scientists of Yale University. Professor Van den Pol is particularly interested in the action of certain viruses on our brain cells. Our brains contain several hundreds of millions of cells. By manipulating the genetic codes of viruses, he can give them an affinity for certain brain cells. The virus is then guided by remote control until it comes into contact with a given cell. But how can the final destination of the virus be visualized? By tagging the virus with genes for fluorescence from jellyfish, Professor Van den Pol can follow the virus's trail through the brain and locate the neuron to which it attaches itself.
There's a lot of excitement these days about using viruses to treat various neurological diseases, but I think the future is going to be very interesting, both using viruses to act as vectors for shuttling genes into damaged cells, and also using viruses to seek out and destroy bad cells such as brain tumors.
Recordings of electrical activity in the treated brain cells confirm that activity has been restored to a normal level.
This was a nerve cell that was infected with a virus, and the behavior of the electrical activity looks quite normal, don't you think? Mm-hmm. The primary function of a nerve cell is to communicate with other nerve cells, and there's a huge number of nerve cells in the brain, and they all release neurotransmitters of some sort. So some diseases are caused by a loss of neurotransmitter, and perhaps the virus can be used as a vector to bring a particular neurotransmitter into the brain and thereby restore communication between nerve cells that have lost their ability to communicate well with other nerve cells. I think as we learn more about viruses and their genetic codes and their behavior inside cells, we can get closer and closer to potentially using viruses as a great asset in the clinic.
The possibility of programming viruses to destroy brain tumors and to prolong cell life opens new horizons in the treatment of neurological diseases. Viruses sent on special missions could become our allies in the combat against cell degeneration due to aging. Viruses could well be the key to eternal youth.